And for this, we have Professor Somdatta, who is currently an adjunct professor at ISO Mohali. I've known Somdatta for over 2 decades now, and our relationship's been a bit like, the class teacher and the backbench kid. So my first encounter was like I'm sauntering down a conference when suddenly someone starts scolding me. I don't quite remember what you scolded me about that occasion, but that relation continues. Most recently, a few months back, we were going to this Indian Academy of Sciences meeting, and as usual, I was in the back bench of the bus talking mile a minute with my collaborator when the front row Somdatta starts saying, "Please don't talk like that loud in Bengali." So you can see what kind of relationship we still continue to have. So it's almost like, I'm still a kid to Somdatta. And it's been a pleasure to learn a lot of things from her in the middle of all the scoldings. And she's probably one of the foremost, probably a pioneer in mathematical modeling in India in biological systems. And it's a great honor and privilege to have her speak in this workshop. This is, of course, not her first time at IMSC. She has been here many times, and every time have given wonderful talks on, how to use mathematics to understand living systems. And so without any further ado, Somdatta.
Okay, thank you, Shtabra. Thank you all for being here. Every time people say such things about me, I don't know whether I should take it as a compliment or as something, oh God, what an awful person you have been. But since they still keep coming back to me and ask me or call me, I hope it's not that bad. Okay, there's one thing I would like to say that since last 2 years, since I had my COVID, I'm a bit short of hearing. So anybody who wants to ask me a question, definitely ask me, but do use the mic, okay? Because that will be very useful. I'm having difficulty in teaching also these days because some students who sit at the back, they don't speak loudly. Okay, so I did tell Stavro not to, call me to give a talk here because it's for a while that I've worked in the area of developmental biology, even though that's where I started my life with, and I'll show you one or two slides of what I did in that older time, because that will give you an idea about how things have changed. Today morning you saw those pictures which Shashi said are green, blue, red, all sorts of things, fantastic microscopy, ultrastructures and all that. Whereas we worked at a time when none of these were available. So how did people go around doing that? Because all the concepts of developmental biology, pattern formation, morphogenesis concepts have come much before they were proved experimentally. So thinking-wise, you really don't need those red and greens. To prove what you are thinking, whether it's correct or not, you need those red and greens to show. So I'm going to give you a little bit of view from those days and also try and tell you what kind of, what is it that people thought about those days and what is it that my ideas are about designs. Now, you may have noticed that I did not say pattern formation in living systems, which is what this meeting is about. I said designs. Okay, now I'll try and tell you why I called it design and what I think are different. So the other thing in our time was that since you did not have so many experimental tools in your hand, you thought a lot more. You thought about using words. You thought about, will this be applicable to that? What if this is not there, etc. So there are a lot more imagination to be played with rather than very specific data that you collect the data and then you work on that. Along with ISO Mohali, I am also associated with ISO Kolkata. I teach a course. It's a course which I started at ISMHali called Theoretical Biology. I just completed giving that this semester, and one part was on pattern formation models. Okay, let me set the scene with lots of pictures are there, no calculations because that you will do. So let's look at something which is there from the net. If you consider the entire evolutionary time on Earth since the Earth came into being, if you consider the entire time of 4.7 billion years in 1 hour, then what is known till date is something of this sort. Within about 10 minutes, people inferred that the life originated, okay, within 10 minutes of the Earth crust forming. Even though the first bacteria is about 17 minutes or so, people think that it has come even earlier through the fossil evidences. But if you look at it a little more carefully, you would notice that about 3.8 billion years ago, or this 8 billion years ago, the first cells with the nucleus came around 41 minutes, algae and seaweeds. The first multi-celled organism with which we are interested here came just about 20 minutes past, okay? These are multi-celled organisms, but they really don't have a great shape and designs. It's within a fair amount of time it took the blue arrow, and you can see the last red line where there are lots of lines, which means between 41.45 minutes to that point, not much happened. Even if a lot happened, there is no proof. What happened at that time? This is what happened at that time. Or they are given like that, but actually everything happened only. You can see even this one, they are all connected to this. So a whole lot of stuff happened after the first multi-celled organisms came, and then you can see the fish, land plants, amphibians. So every time you look at this, a picture comes to your mind. When you say reptile, you know how it looks. When you say fish, you know how it looks. So there's a design, a structure or a pattern that comes to your mind, rather than when you look at a seaweed or algae, something like that. So as you can see that in the last 10 minutes or so many structures have come up. Okay, there's an interesting paper which came. So all these are vertebrates, okay, fish, land, plants. Then there are invertebrates also, which have gone on. Invertebrates means those who have a spine, which is giving me a lot of trouble now. But those are the ones. It's a completely different body plan. Okay, you have to understand that having two cells sitting next to each other and having, as Shashidhara was telling, 10 to the power 9 cells in a complete structure, it requires a lot of effort. For a lot of effort means a lot of time here to come up to that. But there's a paper which came out recently which said that we came about less than 1 minute, and we think we are the ones in this world. There's a paper that came in 2014, 24 on 15th April, which was just 2 weeks back, in Nature, Ecology and Evolution, where they found an animal which actually seems to be the last common ancestor of vertebrates and insects. Okay, so this is a very interesting paper. Those of you who are interested, you might look at it. And what this paper says is, about 700 million years ago, through a whole genome duplication event, an animal emerged which had a front and a back, a top and a bottom. Okay, having that itself is a huge, body plan innovation. And, there are words like innovation, you'll come across slowly, robustness, all these kinds of words which will keep coming with patterns and body plans and all that. So this innovation of having a front and a back, a top and a bottom, this happened about 700 million years. They worked on 20 organisms, and they said that this groundbreaking adaptation at the time, which laid down the basic body plan of the most complex animal, including human, came up. And that's why it's called the last common ancestor for bilaterian supergroup of animals, which has both vertebrates and invertebrates, like insects and all that. And they did a huge study. About 7,000 groups of genes can be traced back to the last common ancestor to bilaterian. Now, what is interesting here is this started with a whole genome duplication event, which means whatever it had, it just replicated the entire whole genome. Then it kept one copy for whatever job it was doing, so that stayed. With the other copy of the entire genome, it repurposed. These are all things, even it's difficult to even conceive how one can take a set of. So this they did by comparing the genomes, etc. And they saw that all these different genes in that original duplicated genome at different specific tissues in the body, it got repurposed to do certain things. Some of them went to work for the neural system, some went to work with the muscle system, etc. So these are kinds of papers which keep coming, and some of us who have have a very wide interest, because I started working in an area in a time when people were very interested in having a wide general idea about what's happening in science. It was possible because there were no 2,000 journals to look at. You still looked at most of the journals. Now it's almost impossible because number of people who work have gone up, details have gone up, etc. But it's always useful to look at somebody else's area, because you always learn something and you learn to use it in your area. I have done that many times, learning from somebody else's area and using it in my own area. Okay, so here is something which I thought I would start with by saying that, this body plan stuff, or the pattern that you see around us, there have been very specific times where this kind of changes, adaptations have happened, and they have been considered to be very major adaptive events. Okay, so one has it's still coming out. Nothing is final yet. So many questions are still not understood. Okay, let me say a few words on what I mean by design. Is there any difference between design and pattern? Okay, most of it is what I think, so you can take it or ask me questions or even think differently. On the whole, they seem to be the same, and I'll be using the word pattern more regularly because it's so common in biology, especially in developmental biology. But I still want you to think about it, the differences. They can have very similar meaning, they can have different meaning based on the context. So we know that pattern is what? If somebody asks you to define pattern, you would say it's some kind of a motif which recurs. Okay, so it's a recurring or repeating theme, structure, or arrangement. It's supposed to be elaborate, complex. The details are much more. Scale of applicability is there, and it has colors, motifs, details, etc. That's what you look at the skin pattern. You look at the seashell patterns, and you'll see how beautifully they come up with it. Then what is design? Design, on the other hand, refers to the intentional planning and arrangement of elements to achieve a specific goal or purpose. So what does that mean? Which means it's a much broader term, which generally you can think of like a plan, a sketch, an outline, a blueprint, or a map, which doesn't really show you exact details, but it's a class of patterns that go into a particular design. Okay? That is what I look at it like that. That design is a much broader term. A lot of patterns come under designs. I don't know where that is. I have a picture I'll show you later. Okay. So, for example, body plan in animal phylum is a design, because there are some morphological features are common to many members, and also the basic shape, like front, back, top, bottom, etc. So the general structure, there is a general structure which you can explain, but each individual is a little different. So for example, the vertebrates share one body plan, but invertebrates have many. So some of these things like symmetry, layers, segmentation, having a gut, which means the inner layer, limbs, nerves, these are all parts of a design. But in each vertebrate or each organism, it might be slightly different. Now, in evolutionary developmental biology, which is a major area now, a lot of people work in this area. You will be hearing some very good speakers on that towards the end. It explains the origins of diverse body plans. That's what it tries to do. There was a little indication in the morning talk, but I'm sure that will be how has evolution kind of planned it out. Okay. I have also given several references to people who may be interested in reading more, but I'm going to tell you for the others what I mean. Okay, when you are talking about living systems, I talked about designs and living systems. So you obviously have to first define what is a living system, which means what is life. This morning also it was told that there's no all-encompassing definition of life. Different people have looked at it in a different way and explained it in a different way depending on from which background you come. For example, this is a very common definition. It's a self-sustained chemical system capable of undergoing Darwinian evolution. Okay, so all these names, some of them you have heard, some of them you haven't. If you haven't, please make sure that you have heard these names, because J.D. Bernal, Eugene Wigner, John Avery, these are very well-known scientific philosophers also. So Watson and Crick, of course, you know through the DNA structure, but these people are also as original thinkers, in fact, more original thinkers than these people are. Okay, so, but then one wants a definition with which you can use it. So when the first US aircraft was going to Mars, so NASA asked James Lovelock, I hope some of you have read his books. In 1964, he was asked to develop a theoretical life detection system to look for life on Mars during the space mission. So what is it that he would do? So he, being a physicist, and of course interested in biology, at one time every physicist was interested in biology, simply because it was not easy to explain. He said, I would look for entropy reduction, since this must be a general characteristic. And I think most of you know that why in living systems are essentially pockets of lower entropy than their surrounding. So that is something which, if you look at the laws of thermodynamics, living systems actually go against it, because it is making order. So if you start with cells which have lots of chemicals and all that, they are put through different types of chemical reactions and etc. And nucleic acids are made, proteins are made, cellular structures are made. So whole lot of definitive structures are made out of some simple molecules. Okay, so obviously it gives you lower entropy than the surrounding. So he said, I will look for an entropy reduction system. Of course, I don't know how he planned it, but that is what he said. But Schrödinger, of course, as early as 1944, he said that in his very famous book, What is Life, which had influenced almost everybody at that time, he had said that living matter, while not eluding the laws of physics up to the date, whatever was established up to date, it's likely to involve other laws of physics which are still not known, which will be revealed and will form an integral part of science as the former. Now think about it. did we really find more laws of physics from 1944 onwards? Think about it, because the whole area of nonlinear dynamics came up much later, okay? And everything that had to do with, systems which are chaotic, systems which are fractal. Those kinds of systems were not really known. There were no laws given in physics till then. So there's still, I'm sure there are things which we still don't know, because we still are unable to, measure the complexity of a brain, let's say, and whether that kind of density of neurons, whether when they are put together in a small space, whether it elicits any kind of behavior which we still don't know from very highly condensed matter, we'll have to see. Okay, we don't know, but we'll see. Yeah.
Would one say that there are any new fundamental laws? I would argue there are none.
Well, you can argue, sure, because I don't have any proof that somebody has found a new fundamental law. That way, there is only one fundamental constant that has been found in 20th century. Do you know what that is?
Oh, H cross.
No, not H cross. The Feigenbaum constant. The repeatability of period doubling. That is a constant, and that's the only constant that has been found beyond Planck's constant and all that. So things do happen. Things we don't know. Maybe not, maybe, but we should be open. I mean, as a biologist, being in a biology department for 10 years, I know how difficult it is to give any open idea to them, something which you can't test immediately. But things, depending on how well you can measure, how fine scale you can make your measurements, things are coming out. We have to see those things. Okay? It's quite possible there isn't any, but let's keep things open. Okay, so if I can't give a specific definition of living system, how do I describe it? So there are some properties that has been given to living systems, and these are, you have heard all these things: homeostasis, it will be organized, it should be, structurally composed of one or more cells, it has metabolism which gives it energy, it makes energy, breaks it, there's growth, there's adaptation so that it can change over time in response to, and of course it should respond to stimuli in reproduction. So these are part of physiological functions, and they have, obviously, I would like to believe they have underlying physical and chemical basis, signaling and control mechanisms that are essential to maintaining life. And you can see the area that you are interested in this meeting, in this workshop, actually covers almost everything, because your structure should be such organized that it best fits the environment that it lives in. So it should have some amount of homeostasis so that it continues to stay. It maintains higher growth. And of course, adaptation comes along with all those things. So this is how one tells that these are the properties of living systems. But living system is a very general word. If you look at biological systems in the world, they are organized in very many different levels. Okay? So we are mostly talking about cells, organs, and tissues, and how they are organized to give rise to a particular structure. Okay? But beyond that, there are much higher orders organized systems like ecosystems, which are made of populations and communities. They are made of organisms. Organisms are made of organs and tissues and so on. At this point, if you break a cell, you get all these. Okay? But it doesn't retain any, you can't go back. Okay? So once you break a cell, as we were talking about, that's the smallest unit of life, or whatever that other word he was using. So if you can break a cell, let's say this is a picture of a cell, so obviously not reality. So it's a model of a cell. If you break it, you can take part of these things out. Some of them you can't break further. For example, if you break a mitochondria, you can never put it back. So some of these cellular architectures, intracellular architectures, People have not been able to reproduce them, make them again, because they are basically, when a cell divides, the template to template division happens. So there's a lot of interesting thing that happens in the cell. We may be only interested in the mitotic spindle, because that basically divides pulls two nuclei out after it's divided. But if you want to know how is a mitochondria made with such a beautiful structure in it, nobody knows, because it's never made. It's actually duplicated inside the cell, and the structure itself transfers from one to the other, one cell to the other. Okay. So what do we get? If we break up a cell, we get a part list. Okay. So this part list will be able to tell you what all is there, but it actually doesn't tell you how it works. So essentially, if you give a car as a toy to a boy, it will break up to find out how it works, and finally it can't put it back together, so it can't break. It can't make it work. So it says, I took it out to see how it works. Now it just wouldn't work. You may say that, okay, we probably don't know the connectivity as well here. Here, if you really know, but mechanics should be able to put it together. But inside the cell, we still have not been able to make a cell given all its internal bodies. And that makes the synthetic approach still not feasible. The reductionist approach has given us a lot of information about all these levels. With the synthetic approach, we haven't been able to do this. Because we know how to, as in tissue culture, we can keep a liver. In a tissue culture, we can make other tissues system in vitro, as it's called. But we have not been able to make a cell yet to make it a living cell. So when you look at any level here, the behavior at this level or here is determined by the microscopic interaction rules involving the lower level constituents, which means how does the organ and tissue behave depends on what cells they are made of, what are the properties of the cells, but it's more than that. Okay, so it's determined by the interaction rules governing these lower level constituents. So it's a multi-level and multi-scale description. So what do? People break this up and take out the tissues. For example, you can take a muscle, get the muscle cells out. You can take a heart, know how many types of heart cells are there. But it is still not, you still don't know how is it that you got that organ from here. All right. So basically what I'm trying to tell you is that each of these levels you can consider as a system. Okay. So the system is a whole. When it works as a whole. Okay. So it's not only a structural whole, it's also a functional whole, which is whole with W, which is made of lower level entities which interact according to certain rules and patterns, can also modulate the constituent entity's behavior. So this is where, when you are working on systems biology or something like that, you may know the parts list, you can put them together, and you might have some idea about how they are interacting with each other, but still you are unable to get the whole thing working, because we don't know how they are positioned with each other, which comes before what, and things like that. So these rules and patterns which are actually among the interacting subsystems, that is very important to understand the whole. Okay? So it is just not the information as the parts list, you also need to know how is this information regulated. So when you go to gene expression, you'll hear gene expression kinetics, you'll hear gene networks and all that. It's not just to know what genes are there, but it's also important to know which is inhibiting what, which is activating what. Is that an AND gate? Is that an OR gate? How am I supposed to put all that together and then see the expression of the entire gene network? Okay, so it's the communication between the subsystems that decides how the system is organized. So if you're comparing a biological system, let's say an insect, Drosophila, with a physical system which is as intricate probably, even though you can see that all it has is sand, okay, just sand, but this structure has been made out of it. So it's a physical system, this is a biological system. So there has to be difference, because otherwise this will evolve, this will not evolve. So in the major differences, there is a much greater complexity of parts in terms of genes, depending on at which level you're looking at populations and cells and small molecules, etc. And the nature of rules governing interactions among system components. What do you think is keeping these sand particles together? You know what is keeping them together. If it dries up too much, it will collapse. Okay, so there are very few physical properties that we know. For example, viscosity, surface tension. These are things which are keeping it like that. So in biological systems, you have all those physical laws, but you also have physiological and behavioral interactions. You have properties which are genetically controlled, and you also have evolution through natural selection. None of these are basically, these last three, you can see in a physical system. And therefore, when you are trying to compare them, there's only up to a point you can use these forces to explain how do cells move inside a oocyte or a embryo, but there is how it is going to evolve. It requires more information and more laws. So we know that by now everybody knows that the entire information of a structure is kept in the DNA of the in a cell. All cells have the same DNA. It's just how differently they are regulated. So it's information plus regulation. How differently they are regulated makes a hand or a lip or a brain or a stomach and things like that. Okay. So cells, all of them have the same DNA. And the way information gets transferred, you have heard that there is transcription, there is translation, and once there is translation, the proteins are made from the genome, whatever required. They work to have all the different physiological properties like cell cycle, cell division, etc. to happen. And from there, they get together and make all these structures. This is intestinal structure. This is, a wing. This is a two-cell structure, which I'll show later also. These are all multicellular systems, okay? So till this point, they are single cells, what all is happening molecular. Lot more complication with different types of RNAs and all that. But just the information is flowing down this way. You think about the virus, it's just the other way around. Okay. From here, you can go to the other levels. That is, you can make a social system, that you can get many of these together and make a social system, then you can make a ecological system. So many of the organisms together makes a whole system. So some of us who have worked in many of these levels know how similar models can actually be used if you can actually understand where to use them. Now, where is evolution coming here? We all know that the variations happen only at the DNA level. So there is a mutation, something has changed. One A has changed to a C, or a G has changed to a D, or whatever. Okay, once that change is made here, the entire process of through transcription, translation, that change, whether it's going to change the RNA or whether it's going to make a change in the protein, depends on where the mutation has been, change has been made. Sometimes it's absorbed, there is no difference in the resulting protein with the original protein. Sometimes it happens in such a place that the entire protein function changes, therefore it's going to affect this. If it affects this, it can affect here, even though there are other processes here. And then you are, let's say you have a wing, it has a hole in the middle of it because of that mutation. Now this wing is not going to be functional. You know that, the air will pass through the hole and the wing is not going to do the wing's job. And therefore, the fly or whichever organism is having that kind of a wing will not be able to go through the entire life cycle and finally won't be able to fly, and it's not able to produce more offsprings to continue in the system, continue in nature. Okay, so it gets removed from nature. Some mutations which is happening here, and the forces that allow that mutated system to continue with this whole life cycle is something which decides. So adaptation happens in all these. It depends on the environment. In all these places, mutation happens there. So this entire cycle is very important for any mutation to continue in population or not continue in population. So to keep your mind open to these things, every mutation does not. There are names for such mutations. Even if you don't remember the names, just remember what they are doing. So when we are talking about designs in living systems, there are two processes that in biology people talk about. One is pattern formation, which self-organization comes under that. So it's, I said that it's a formation of regular or logical form, order or arrangement of parts in space. So things like coat marks, heartbeats, calcium waves, these all come under pattern formation. Basically, these are happening without too much of physiological, for example, without much of cell division, cell movement, etc. Okay, so self-organization also, it's kind of it develops a form through intrinsic interactions. I'll show you an example. The other one is morphogenesis, and morphogenesis is actually involves orchestrated cell division, movement, and differentiation. Okay, so here you have structures coming up. From near homogeneous initial condition, you have a structure coming up. So when I started in this area, in books, first of all, the books were not called developmental biology. Books were called embryology, and there would be a chapter in frog, one chapter in chick, one chapter in something, and all those. Now you go and look up a developmental biology book. Initially, the initial chapters are all in initial common processes during development, which is common across all organisms. This is how field changes. From very specific information, then you find out general rules. And then, okay, in frog this thing happens, in chick that thing happens, etc. and all that. So, in those days, pattern formation and morphogenesis were separated using these kinds of things. They're still to some extent done, but a lot of patterns require morphogenesis, so they are more or less mixed up now. So when you are looking at morphogenesis, you are looking at limb generation. This is your lung tissue. This is the whole insect. Whereas here you can see that it's a coat pattern. There's a scaling, the scaling laws in these things. In fact, morning there was discussion on if you take an embryo, right in the beginning, the bicoid has bicoid mRNAs on one side, and then the bicoid protein is made, and then it diffuses. So there was some question which I thought could, a little more information would have been good. At least till 12 hours or so, if you take that embryo, cut it into half, you get two embryos. Smaller in size, but exactly the same. So there is a scale, I won't call scale free, but scale independent phenomena that goes on in the initial parts of the pattern forming system in these embryos, but beyond a certain point, once I think after 12 hours or so, you can make it 4 parts, 8 parts, and you'll get small embryos initially. But towards after a while, then all these gradients are set up very well. Other proteins, all those reds and greens come up, and therefore the plan is much more laid out than initial period. So there is scale invariance, and that's the work of somebody called Spemann and Mangold, whose paper came in 1924. This year is the 100th anniversary of that paper, where they call that bicoid. Bicoid was not known at that time. They just did transplantation studies and showed that whatever is there on that initial end where the sperm has come and entered the egg, that part is called the organizer. And there's been beautiful experiments by Hilda Mangold and Hans Spearman. Hilda was a PhD student of Spearman. It's known as Spearman's experiment, but it was the PhD work of Hilda's. So that paper actually started the whole area of organizing activity of these initial some of the gene activities. Later, of course, the entire homeobox gene system comes up, and then it becomes further organized. In self-organization, most of you know, self-organization is studied using bird flocks, bacterial swarms, fish schools, animal herds, where you have a large number of individuals which stay together and they move together like a unit.
Ma'am,
Yeah.
I had a question in the
Yes.
previous slide. So you said that once you sort of split this embryo, you can get smaller ones, which are they fully functional as well?
So in those days, people could grow them into petri plates, because those days there was no way of having a full tissue culture facility. So what they had shown up to 4 parts, they actually made into small structure which had further made the whole kind of structures of all those different parts.
Oh, I mean,
which
but
came out.
do they like become functional fruit flies that can reproduce?
They do. Functional is a difficult word. They do look like fruit flies, but whether they can be as competitive in the nature, that we don't know. Because they have small wings. They don't grow. And they don't get the size back. There's no size. There is size regulation, but the small remains small. It then doesn't grow to the entire.
Excuse me, ma'am.
Yeah.
Just to clarify, this cutting of the embryo is just after the bicaudal mRNA gradient. Is it before or after the bicaudal mRNA?
So the bicod mRNA, the organizer has to come up, because that decides the polarity of the embryo, which side is dorsal, which side is ventral. And that is actually, I'll show you a small picture, where when the sperm comes and enters the egg, it's decided by a calcium wave which starts from there and moves all the way to the other side. Calcium wave moves, and that basically decides the dorsoventral axis. Now, so bicod mRNA is already there, and when you cut it, so there is the 2 portions again have a dorsal and a ventral part. So bicod mRNA and then the other side, and this one also sets itself up. It is also very nicely seen in some experiments that I'll show on other organisms like Hydra, like how these potential for forming the other structure stays even if you cut it out. This is where the what is it called? The morphogen gradients. If you cut it, then before cutting the gradient was low. After cutting, the chemical reactions grow on, the morphogen gradient comes up in two different in this same repeated structures.
Okay.
Ma'am, sorry.
Yeah.
I had just one more general question. So you use the word design. Many people, we all do in this field. But I just wanted to ask that in the definition of design, there's the word intention, which is typically not there in the evolutionary process. So I just wanted to ask, what is your opinion on this?
So the word intentional was there because here the design is being defined as the difference between pattern and design in terms of making something. Okay. So I have made a general design, and then when it's executed, it's executing in different ways. Now here, when it said intentional, it's obviously not in the biological parlance. It's obviously not intentional the way we think. It's decided by the genetic architecture of it. So unless that architecture is there, you will not get. So initially, even though I have a picture which shows even Turing patterns can give you a kind of, it sets up without any, most models don't even include the genetics in it, but they assume that when I say there's a morphogen, it is actually an outcome of gene expression. That is how it is. Okay, so let's, you can also have patterns produced through extrinsic influences. For example, you can, if the, you must have seen, if the leader says, do this, then everybody does that, some kind of a, so the pacemaker of the heart is one of those, like that itself controls the electrical activity of the heart. Then templates, I give you an example of mitochondria, etc. These are all pre-existing patterns. Okay. Now, I want to give you a few examples of, I have moved to, you can see that I moved to pattern formation. It really doesn't matter. I want to tell you that maybe in developmental biology, you're only talking about form as pattern, but in general, when you model pattern forming systems, they can be spatial pattern, they can be temporal pattern, they can be spatiotemporal patterns. Okay, so I'll just give you a few examples that how do you look for it. Your eyes should be trained in such a way that you look at something and you realize that there is something which I can, study, which looks like there is an underlying interaction rule which is going to give this kind of structure that I have. Okay, so when you have spatiotemporal patterns in biological systems, because they do come up because of nonlinear interacting processes at varied space and time scale. Now, this nonlinear word to biologists don't strike too much. I've realized this. I've been in biology labs without knowing any biology for about now 35, 40 years. And because I myself never studied biology in school and college. But whereas for me, since I came from physics, linear, nonlinear, robustness, all these words are very close to my heart. So, but nonlinear process is at the heart of pattern formation. Linear systems don't form pattern. That is, even in the Turing model, if you look at the Turing model, it's actually the diffusion is, when diffusion is switched off, there is spatiotemporal homogeneous steady state. Okay, you'll study it, I think. But only when diffusion is switched on, which is a highly nonlinear process, the pattern starts forming. So, nonlinearity is a must in any kind of pattern formation. And those nonlinearities come because, there are chemical reactions happening, there's movement happening, there's diffusion happening. Each of these processes are nonlinear. So, molecular reactions during gene expression, enzyme reactions, transport processes, signaling, cell movement, migration, social interaction, gene interactions, everything is, none of these are linear processes. So, obviously, they are very important to have that, to be present in, if you want to have patterns set up in a particular geometry. You can change that through mutations, through disease, through environmental change, and all these are very important to start the study of pattern formation, because each one of them are highly applicable area. So, if you can identify and understand mechanisms that yield specific patterns, you can control the pathology, you can regulate it as per requirement. When you can't do it experimentally, the way we go about trying to figure out, to understand how did this pattern come, is through modeling. So, that is where we can actually change functional forms which are nonlinear. You can change the coupling between cells, feedbacks of different reactions, and different biologically realistic properties that contribute to the evolution and maintenance of pattern in space. So, those of us who have been into modeling, most of us have come from, many of us in olden days have come from physics or mathematics, but these days the courses are such that people do study biology, mathematics, etc., physics in the beginning of their courses, and so in my class, I get lots of biology students doing exceptionally well in theoretical biology. So I think it's very nice to get that, because when I studied doing modeling, I didn't know biology. Now at least people come with good information bank. So if we look at we have looked at this from ecosystem to chemical constituents, these different levels. The structure, function, regulation, interaction, all of them are happening at every level to set up each of these different structures. I'm just going to give one example of ecosystem problem. This is not an ecosystem meeting, but if you look at these, they are so similar to Turing models that you always feel like studying them, because the basic maths and physics is just the same, whether you are looking at an ecological pattern or whether you are looking at a physiological pattern. So this is a structure which is known as the fairy circles. These are round patches of the grasslands of Namibia. So they persist like this for 75 years. And why did they come up has been hotly debated. Okay, so these are created by termites. Okay, these amazing spatial pattern. People have said those who go in rockets, they have even seen it from space. It's that beautifully clearly seen. But what drives it? Okay, it was known that termites are somehow doing, because you can see that the termites live here. But what is it that driving the termites to be like that? So it's strong competition for resources. Okay, and that is if two different colonies come close to each other, they actually fight to death and they separate from each other. How this hexagonal honeycomb-like pattern will come up will depend on the strength of interaction, will depend on the type. If you change the type of termite, maybe it will be different. So these are things it came up. This came out in 2017, because people have been discussing this for a very long time. But in 2017, Corina actually showed that these bizarre patterns in the soil are created by competing life forms. So competing life forms can be competing cells also. So basically, pattern is something which is very general, and you can see it in very many directions, very many levels. For example, let's look at something. I'll go through a few slides, starting from the molecular to slightly higher level, and then I will show you some of my work. Yeah.
So when you say that the underlying mechanisms are the same, in the case of fairy circles, it's not diffusion driven, right?
So it depends how you define diffusion. In this case, it's competition for resources, means there is a certain amount of resource and both organisms are competing for it. So it is not the same expression like you put for diffusion, but it's something like Lotka-Volterra type or something of similar type. So there are certain types of functional forms that you can use. In this case, it's certainly not diffusion, but more like direct interactions, by binary interactions. Even though the pattern that comes up, you can see that it's not diffusion, it's not through diffusion, but it's called Turing-like pattern because it's showing hexagonal patterns that one can see even in diffusive systems.
Ma'am, excuse me, ma'am.
Yeah.
So when we are saying that it is not driven by diffusion, so can't we somehow define diffusion in this context, like a resource, there is a gradient of the resources available in the...
Diffusion is generally, since I just taught this entire course, so diffusion comes under cell movement, okay, or movement of organisms. So what you mean is here also termites are moving, but then you have to have something like a equivalent to diffusion coefficient. Okay, so if you say their movement is something of this centimeters per square and things like that, and equate it to diffusion coefficient, then you also have to have the similar, scalings and similar a relationship that is it changing with squares of the distance and things like that. So you can define, you can say that this is diffusion-like, but it will not be the same expression like a diffusive term. Okay, so here this I'll go through very fast. So even when you're looking at the double helical structure of DNA, you see the structure has come up to facilitate replication, okay. So this was the best structure found, and because it could explain replication, we talked about this in the beginning. So even at that level, not every structure is evolutionary, useful, but this DNA structure which Franz Crick and Watson found was good because it fit the function of the DNA, which is given by the replication. Its replication ability comes out very well. Even when you look at sequences, let's say you want to find out there are so many genes, these are sequences of genes. Even here, when you look at a certain section of the sequences, you see that they are similar. So even this is called pattern actually. All I'm trying to do is using the pattern at very many different ways. Not the, let's say, diffusive instability and things like that, but you can look for pattern, and for looking for each one of them will have a different methodology, theoretical methodology. That doesn't stop you from doing developmental biology, but it's good to be aware of what is also there in the... So, and this being conserved has a very functional role, okay. So, there is a certain motif that it shows which has these proteins have developed functions for. Okay. Then of course protein structures, how beautiful these protein structures are, and each one of them have a certain kind of function that it can do which otherwise is not. And then higher order structures with those simpler structures get together and form even higher order structures, so it can perform certain functions of ligand binding through these kinds of motifs. So the calcium can sit here. So it's a beautiful structure. The people who work on protein structures, I do. So we find these kinds of things very, very beautiful to see that how nature developed it. And at a completely different level, this is the pattern of contacts in a protein interaction network. Okay, you see how beautifully they are set themselves up. This is, of course, has a scale-free nature. So you can look at how did this pattern arise, what is the underlying genetic interactions that has given rise to it, how is it related to the pathways. You can see different types of control mechanisms which have very specific patterns of regulation, which you can figure out from biochemical pathway analysis. You can have, I don't have to say anything here, you have seen this gene expression pattern in embryos. This one gives rise to segmentation, which is a particular kind of functional structure form which gives rise to segments in Drosophila. Epidermal kind of patterns that comes up. Beyond that, you also have something like these segmental patterns in fingers, which has its own functions given. Why is the lung function, lung structure like that? Is it not just like a? Why is our lung not like a liver? That question somebody should ask. What would have happened if our lung was like a liver? Okay, that depends on what is the function of liver and what is the function of lung. So look at the structure, how beautiful. It's a fractal structure anyway, but it just aids in absorption of gas exchange basically. So more surface, more gas exchange, and that's the best way to have that is through a fractal structure. Similarly, when there is decreasing nutrient concentration, if you grow these bacteria under deficient nutrient, they make structures like that, because more structures they make like this, more surfaces they have with very little nutrient to pick up whatever little nutrient are around. If it was growing like they grow usually, like in a blob of bacteria, it would have picked up nutrient only from the outside part. If when there is lot of nutrient, no problem, but when there is dearth of nutrient, this is the structure. So you see, this is the same bacteria if you put them in high nutrient condition, they won't grow like this. So here are patterns which change depending on the environment. This is kind of something which is beautifully shown by the beautiful models also, which are given through diffusion limited aggregates. So this computer is a small computer simulation where you can actually model these things. He's no more, but his website is full of all these things. These structures you all know. They are known for intestine, for nutrient exchange, gas exchange. Each of these structures are specifically suitable for the functions they have, and those of you who are interested in fractals, they should look for it. Sometimes even when the structure changes, for example, here's a mutant plant of Arabidopsis where wherever it's supposed to make petals, it makes anthers, okay? But this mutant remains in nature because it has very high fertility. So it's not that all mutants which change the pattern is going to be erased from nature. It all depends on how well it can contribute to the next generation. Okay, so to change this normal pattern to this altered pattern is some kind of, one needs to know which are the genes. It's known. Say particular mutant. It's a single gene mutant. Okay, temporal patterns. This is a very common example that is given. This is our normal heart rhythm, and then if there is some kind of a pathology that comes up, then it changes its structure, and then it goes to terminal electrical activity. So change from there was a lot of work at one time when I was a student to say why is it that what happens, why is it becoming more regular. So if you do the dimensions of these time series, fractal dimensions, then you would find that the dimensions are reduced here. So why is it like that? So then people also used a lot of statistical physics to use oscillators and coupled oscillators to find out. This is where there were oscillators, they became synchronized, and then it went to oscillator death. So people have gone through a lot of different ways of looking at biological patterns. I'm showing you some of them so that you get some idea about it, since it's supposed to be an overview talk. For temporal patterns, there are other ways, like you can force the pattern. So this is where I'm forcing. I give glucose input into the cell, and depending on whether I give a constant input or a periodic input or different types of periodic input, the output of what I'm measuring here through this, so my input is changing here, I'm measuring through NAD and NADH, they actually change. So you can change the pattern of output by changing the input here. Okay. So this is happening inside the cell. These are some very old experiments done in Germany by people who right in those days were very interested in chemical oscillation. In fact, the entire pattern forming mechanisms, whether it's Turing or whether before that, it's from people who were interested in chemical oscillation. Because, chemical oscillation, chemists have been part of this area for a very long time, and whole history is there of how different ways they got into to show that we can, even Turing's model is a chemical system. It shows you that what happens when two chemicals interact with different diffusivity, interact and move. Okay, all right. Here is a spatiotemporal pattern which you can see that how it works. So how many people don't know this? Cellular slime mold aggregation. Is there anybody who doesn't know this? If everybody knows, I'm going to not describe. Okay, so here is an organism called cellular slime mold. I think you're going to hear more talks on this. So when they get to eat a lot, they live, as they call, asocially. They basically, in the Petri plate, They just eat and move around. They don't care to talk to each other. They don't even know who's around them. As long as they get food, which are E. coli bacteria, they grow, divide, and the moment you stop food for them, it takes about 8 hours or so. These are my early experiments when I just started moving from physics to biology in Bangalore, ISC Bangalore. So what they start doing is, some of the cells, they start becoming centers and other cells go towards it. Okay? And then they start making these beautiful structures like this, and all the cells come to the center, make a mound of cells, which is called a slug, and then a whole lot of developmental process goes on, and then it till up to some point they are just a single amoeba. At some point between here, there's differentiation, and there are two types of cells that come up. One type of cell forms the stalk, the other type of cells form the spores, and they stand up like this. This generally happens when the environment is bad and there's no food. So some cells enclose themselves and stay as spores. When the environment is better, they break up, come out, and they go back to this. And here is a whole lot of work that has been done in this area, because this shows that in the Petri plate, through a small tube, the experimenter is releasing a chemical called cyclic AMP, because people have found out why is it that they go towards it. And all the cells around are experiencing that cyclic AMP because it's diffusing around, and they move towards it. So they receive the cyclic AMP, they amplify that cyclic AMP signal within themselves, then they move towards it and release the cyclic AMP signal for the further away. At some point you will see this tube is taken away and there's no more cyclic AMP signal is giving. And what happens to the cells? You can see the tube is gone. They are not getting the signal anymore, and therefore the cells are moving away from each other. You can see that they are no more. They don't feel this. Now, there's a whole lot of, there's beautiful models and whole lot of work on this. So this is a spatiotemporal pattern that is developing due to certain lifestyle changes. For example, no food. Okay, what it tells you that there is active communication and interaction among the cells in a population, which leads to the pattern. So that starts from here, and then it goes here, and finally it goes here. Okay, so this communication among cells and interaction among them is, and of course cell movement, that is prerequisite of the form of the pattern. Sorry. I'll just I'll pass through these. These are activity patterns. So these are biological rhythms. So free running activity of mouse. So when you put them into a 24-hour period, they actually repeat the activity on 24-hour period. The moment you release them from that 24-hour period, that is, keep them in area where there is no either complete darkness or complete light, then the entire activity period moves away from that 24-hour period. There's again a lot of models and work in that. Okay, so let me, this is what I was talking about. So here is that once in a frog egg, once the fertilization occurs, this is what happens. So this is where the fertilization has occurred, and a calcium wave actually goes from one end to the other end, basically setting up the dorsoventral axis to it. Okay. Anyway, what I'm trying to show here is to have any kind of pattern to set up in a multi-cell system, a system where there are multiple cells, cell-to-cell communication and cell movement. These are two major processes that are required. Now, cell-to-cell communication, there are a whole lot of different ways cells communicate with each other. There are, gap junction, there are hormonal type of interactions, there is receptor ligand binding, here is a molecule moving from one cell to another cell through a through when two cells join together and make a whole between them. And then there's if cells are sitting on a matrix, then through the matrix also cells can communicate with each other. This is known, this is textbook. What is not known is there is other kind of contacts which are long range, transient, these and a nearest neighbor also. So I have given just one example where these are two cells and the distance between them are multi-cell distances, and there are processes, these are called processes that connect these two cells through which they exchange intracellular material. It's not very common, but in developmental processes this is very common. So this is a paper from developmental biology. There are several of them. These are called long range connections to transfer information from. So when you're trying to model them, basically when you have multiple cells coming put together, then they have to interact with each other. They can interact only with their nearest neighbors, but they can also interact from with some cell which is sitting somewhere else through these long range connections. And some of you who work in this area, you know how this nearest neighbor interactions are actually make the type of interaction makes a difference. So here is a picture which you saw in the morning. Here is the kind of gastrula, which is the cells are moving, they are coming inside and distributing themselves. And this is the most important event in embryogenesis. If this is defective, embryos don't work. So that is why Lewis Wilpert has said that nothing is important but gastrulation, which is the most important time in your life. That is, it sounds funny, but that is how it is. So cell movement, which is shown here, and cell-cell interaction through different types of contacts that are shown here are very important for any kind of cell pattern formation. And the way different molecules or cells or things move are through the fluxes, and these are three different types of fluxes that one actually models even when you're doing Turing or you're doing slime mold aggregation. You just have to see how the cells are moving. If they are moving, they are not moving, but the cells are in a fluid. For example, blood cells are in blood. The blood moves, so the cells move. Cells don't move themselves. So that is, you have to write transport equation for them, mass transfers. Diffusion, of course, random motions of the particles or the cells in any substrate or in a fluid, which is very common. And then there's field equation, which you can use to model phototaxis, chemotaxis, etc. These are three major fluxes which you use for modeling cell movement. I just mentioned this because I thought it would be useful for you to know this. The other thing which is very important is geometry. Okay, so when you are working on with a set of cells, they're confined in a particular geometry. Now the boundary conditions or the way the cells are arranged with each other is very important for the pattern to arise. So you can have, this is the Arabidopsis root. So you can see that you can basically we model it with a circular geometry. So I have j plus 1, j minus 1. So if this is j, then this is j plus 1, j minus 1. And after n cells, it repeats. Okay, you can have fixed boundaries also with this. Basically, those of you who have worked with this, you can also have, this is the Drosophila pupa stained for 3 different dyes, and you can see the cells are stained like this. You can have this kind of honeycomb like 2-dimensional arrays. So whether you are trying to model in 1 dimension or 2 dimension or even 3 dimension, you need to choose your geometry properly. Okay, so basically the kind of theoretical approaches that one uses in these different levels of biological organization. For example, when you are studying information in nucleotide strings, for example, DNA, RNA, etc., you use probabilistic methods, dynamic programming, set up algorithms, probabilistic algorithms, or you can also use Boolean models if you're working with gene networks. I think you have classes on Boolean models. Then if you're looking at protein structure folding, you can use all these different methods, molecular dynamics to networks, everything. Biochemical pathway, of course, dynamical systems theory, networks, differential equations, Boolean algebra, topology, all these kinds of different theoretical methods one can use. Tissue pattern formation, differential equation, partial differential equations, couple map lattices, agent-based models, graph theory, topology. So anything that you are interested in, you could use it. Ecology, epidemiology, of course, very similar. So if you are working in this area, those of you who want to do theoretical modeling, you have to know some amount of how to use spatiotemporal systems. The easiest one to do is couple map lattice. You can also use partial differential equations. You can do it if you know how to do it, and differential equations, graphs, etc. So some of you who are interested in theory, you can understand some of these or learn some of these. Okay. Now I want to talk about a bit of history in last 15 minutes or 10 minutes, whatever. Here are some pioneers. What you see today in developmental biology is based on the work of huge number of people. Many of them are not really physicists or mathematicians, but they are very interested in theory. Okay? So I haven't gone beyond. Actually, the first biologist is Aristotle. Okay? So I'm not going to start from there, because he's the one who was not only interested in philosophy, he was also interested in biology, especially he was interested in embryogenesis. Embryogenesis has always attracted people because it's very, very interesting to see a whole thing coming out. You start with a single cell and then a whole baby coming out or a whole lizard coming out. It's just incredible how it happens. Okay, Weismann I've started with. He had a theory of germplasm, 1892. It's interesting. By the way, a whole lot of them are Germans. I don't know whether this is biased or not, but at some point in those days, Europe was the hotbed of academics, and a whole lot of science was done in Europe. And in Europe, Germany, of course, was very, very active. Now, of course, when you want to look at the experimental work, you see papers from Japan, from USA, from other places also. But these are all, you can see they are all Germans. And one of the major centers for embryology was Freiburg, okay, which is where Weismann worked, which is where Rooks worked, I think, I don't know, Spemann and Mangold worked. So there are schools which only study these things. And if you are interested in history, then it's very satisfying to see who learned from whom, what was the social structure at that time, what was the most important or the prevalent philosophical thought in those days. A lot of them actually follow Kant, so Immanuel Kant, but that's because I'm interested, so I read all these things. I hope some of you would be. So Weismann talked about theory of germplasm, which was very interesting because he believed that differentiation and development results from the specific partitioning of genetic material. Nobody knew what genetic material, nobody knew DNA, nobody knew genes. But through experiments, people felt that there is something which is transferred from generation to generation. Hans Driesch, he was a very interesting person because he had the analytical theory of development, one of the great and influential books in history of developmental biology. So he actually started his research to explain development on the basis of physics and mathematics. Self-regulation, scaling during development. All these are very, very interesting. All they would do is cut the embryo, see what happens and things like that. He also introduced the concept of positional information 75 years before. Wolpert did it in 1969. So sometimes we reinvent things. So positional information is one of those. Rooks, who is very, who Ernst Mayr actually said that after Darwin, he is the most important biologist in the history of biology. So Wilhelm Rooks, he coined the term developmental mechanics. There was no experiments were all very kind of mechanical experiments, and still they had a lot to think about, that how is self-regulation achieved, how is development a scalable method. D.R.C. Thompson, his very famous book on growth and form, 1917, he was very influential because everybody wrote this book. I don't know, I'm sure the library has these books, but nobody today looks at these books. I used to have a copy, I don't know where it is now. So some of these are very, I hope someday somebody, Google must have digitized this. Spivak and Mangold demonstrated in 1924 that animal bodies develop from a patternless single cell rather than growing from a microscopic preformed version of the adult body in humans. So there was something, a concept called homunculus basically, which said that actually inside the egg there's a small human shape hiding there, and all that happens is that human shape becomes bigger. It's a very weird thing, but that's what most vitalists and most of these people believed. They didn't believe that pattern can come up from nowhere. Okay, now that nowhere is physics and chemistry, but they didn't believe that, so they demonstrated that pattern-based single cell, you can develop animal bodies by doing through. They demonstrated that, and it's not that there is already a small thing sitting inside which actually grows. Okay, Waddington, very well known. His book on organizers and genes, 1940. Again, 1940, genes were there, genes were coming in, but not as much as you see them today. People could not do as many experiments. Molecular biology was just coming up. So he was very influential. And I think Waddington's landscape, developmental landscape, developmental fields, you will study, somebody will teach you. For us, the paper which is very, very influential is 1952 paper by Alan Turing, which came out in the Philosophical Transaction of the Royal Society, which is called The Chemical Basis of Morphogenesis. Now here is a paper which I cover in my class. It's an interesting paper coming from a computer scientist, that how, if you are interested in Alan Turing's life, you should read. These were all very colorful people, worth knowing how what they went through and how unhappy they died. So his focus was on chemical patterns, because there was a whole school of chemical oscillations and patterns in Europe which was working at that time. Turing was in England, so he coined the term morphogen. So they didn't know what a morphogen was, but it was a molecule capable of inducing tissue differentiation. We still believe that, but now people can show it, and those days nobody knew, but people thought these are. So what you see today being shown experimentally are just things which have already been talked about earlier without knowing what they are. So the Hox gene cluster, today morning you heard about they are actually the morphogens in Turing cells. Here is another set of experiment chemists. People don't talk about them a lot, but Prigogine and his co-workers, theoretical chemists, actually they extended the non-equilibrium thermodynamics to situations far from equilibrium. So they talked about how structure or pattern can arise from almost smooth, not completely smooth, almost smooth background concentration variations. Basically, at the heart of pattern making is symmetry breaking. We all believe that. So these are all Turing patterns. So here are the different, when you model, this is how your concentration of morphogens will change. and those can be translated in 2 dimension into these kinds of patterns. Okay, so you could call these as kind of designs and these as patterns, but even within the same, just by changing geometry, you can see the same organism will have dots, will have radial things, will have stripes here. So simply through geometry. So this is one area which everybody is interested in. Now this is my lifetime and very well known, Girer and Meinhardt. So Girer started, both physicists. He started his group in Tübingen, again Germany, and then he shifted to developmental biology from physics with Hydra as a model system. And then Meinhardt joined his group and they were very influenced by ideas from cybernetics, because cybernetics school was just next to their school. And there they were studying contrast. How can you enhance contrast through what kind of algorithms do you use? And Girer actually, and both Girer and Meinhardt got very interested in that, and they used that to formulate their theory of local self-activation and lateral inhibition. So what they did was they combined local deviation amplifying processes. So there's an autocatalytic process with lateral inhibition. Okay, they explain structure formation starting from a homogeneous state. So the difference between Turing's model is that they included nonlinear autocatalysis, the activator, and also differences in diffusion rates of components, which is part of Turing's model. So this short range activation with small diffusion constant, the long range inhibitor action for larger diffusion constant. So this is very common. And somebody in the morning was asking, why is bicoid mRNA only localized to a small area, whereas protein moves that? They just have different diffusion constants. That's all. mRNA molecules are much larger. They are basically much larger molecules. They diffuse very slowly. Okay, so there's a book which Meinhardt wrote later. I bet the models of biological pattern formation. It has so many examples. It has just too many examples. Okay, just to give you a small background. So I started my PhD in 1979 or 70. Forgotten. So, and it's that old. So Girer-Meinhardt model was then at that time. Turing model was always there. And I decided to join a group which works on Hydra. So Hydra is an organism which is very interesting. It's a slender, long, one-dimensional organism. And if you cut its head out, it will grow another head there. If you starve them, so this is how they reproduce, through budding. Okay? So if they're starved a little or given less food, they don't bud. But they stick on the glass plate through what is known as the foot or the basal disc. So they look very beautiful. You have to keep them in water and you have to feed them, clean them twice a day. It's almost like taking care of children. So I started working in this area and I did some experiments. Absolutely no molecular biology in those days. The kind of experiments I did, I'll just show you. So there through our experiments, we built a model called Hydra pattern is controlled by two distinct but interacting morphogen sets. We also, so this is a scheme of activation and inactivation. So there's a foot activator, head inhibitor, head activator, foot inhibitor. So there's a cross reaction which is activating and inhibiting. This is the scheme of interaction through the experiments that we did. That I did, and I'll show you the experiments, just one slide. And that's what we published in developmental biology in those days, perturbation morphogen gradients induced budding in Hydra. So what didn't we do here? Very simple. I'm showing this to you at what level we had to work at in those days, because there was, the maximum molecular was that you put some radioactive thymidine and see whether it's incorporated in the DNA or not. That is the maximum experiment you can do. Okay, so here is the Hydra. Okay, so you take this underwater, look through the microscope. They are some 1 or 2 millimeters long. Then what you do, you cut it with a knife, some small part of it. The problem is the moment you touch it, it becomes a round ball. Some of you have seen it, you know. So doing this took me 3 years, just to be able to keep it straight and then cut a small piece of tissue from there. And then this is the size of the tissue, this is the hydra. Okay. So then what do? You make 2 parts of the same tissue, and then what you do is you make it 2 parts and transplant a small part into near the head in another hydra near the foot. Okay. Now what do you expect to happen? This is exactly what Mangold and Spearman did. They just cut pieces, transplanted it at different parts of the embryo and found out what was the result. So what is the result? It took me 8 years to do my PhD because first 4 years I didn't get any result. First of all, I never did biology after class 8. After 8th standard, I never did biology. I was first time doing it during PhD, but it's okay. So when you do that, what happens is the small tissue here, within about 24 hours, it became a foot. Okay, and this one became a small head here. So if you look at them, here is the foot. Okay, so it will go and fix itself on the bottom of the plate, and it will become like a. So you could see that this has become a foot, and you could see the other one. This has become a head. Okay, this was the main experiment, but doing this entire procedure was very, very painful because it was first of all because it's completely hand-on. You have to do it in 100 or 50, 100 of these cells. Okay, I think I'm going beyond my time. So how do you explain the results? So we did lots of other results of transplanting them at different potentials at different. So again, there's a gradient of morphogen around it. So you transplant at different points and see how many you get and all that. So we actually simulated the experiments of this mid-piece grafting in a simple Guillen-Meinhardt type lateral inhibition model. But we added weak cross reaction between the head and the foot inhibitors, and the chimera showed 2 foot activator peaks at the 2 ends. Okay, so here is one foot, here is another foot, but it also showed a head activation peak here, which we didn't get. Okay, so it just says that this model cannot explain the coexistence of 2 terminal structures unless constraints are put on the ranges and details of interaction between head and foot specific morphogens. So this is the kind of experiments one did. One also did, like Spearman and Mangold also did. They took children's hair and they took a piece of tissue and passed the hair through that, and then they'll keep it because then only they'll stabilize, and then they look at what happens to the tissue after that. So such kind of experiments I've done many. So basically, this is what I'll stop here. Let me just go to. Okay, so let me stop here, and thank you very much for sitting through this long talk. I don't know whether I've been able to give anything new to you, but I just wanted to give some kind of a feeling to you that what this field involves for both physicists and mathematicians and biologists. So biologists these days are very smart. They are as good in maths and physics as several physics students. My ISA students are very smart. They also learn biology a lot. So this is the right time. The people who are trained in both areas partly and does what they like most. Read a little bit of history, read a little bit of philosophy, and of course you will do maths and things on your own. Read biology. How history influences present day biology is also something you should know. How social structures influence. These are all very interesting topics for you to start worrying about. Thank you very much, Shitapro.
Thank you, Somdatta Di. So that's been a long day. I guess people are really tired. But you're around for a few more days. So, if they have any questions, they can always catch you.
I'm here tomorrow morning.
Yeah. You're leaving only on Thursday, right?
Day after tomorrow.
Day after tomorrow, yeah. So she'll be around. So in case you have any questions that come to you, you can catch her during lunch or dinner and so on. And Mithun will actually be giving a full-scale theory of the Turing pattern formation that Somdatta mentioned. So you can basically have a full-blown analysis of this thing. I think just wanted to point out though that this diffusion that you were saying, I think more than the diffusion is the nonlinear part which actually is responsible for the pattern formation, because diffusion by itself would just cause homogenization.
That's what is Turing's.
Yeah.
The most important thing Turing's model did was to show that otherwise diffusion homogenizes the system. But in this case, along with the chemical reactions, diffusion actually sets up patterns. So Turing's work is special only for that he used diffusion to set up patterns.